Hello class. So today we'll be talking about basic methods for microbial enumeration. So what is bacterial enumeration? Sorry. Enumeration of bacteria is basically trying to count the number of bacteria you have in a given sample. This technique is mostly used in food microbiology and dairy when scientists or lab individuals are trying to calculate the amount of bacteria they have in a particular sample. It's basically important to know the food safety, quality of the food you're trying to consume, to know the level of organisms that are present, to know if this food is actually good for consumption. So we'll be talking about different methods in which we're going to be using to calculate these organisms. And the first method we'll be talking about is this direct cell count. This process counts number of bacteria in a liquid medium or colonies on a plate. It probably uses the counting chamber. You can also directly counting chamber using the microscope. There is something we call the Petrov-Hafel cytometer. That is what we actually use to do this. So this is a pictorial view of it. And I'm trying to give a PowerPoint a point of the procedures that we use. We use a capillary pipette to take a drop of the broth culture on a slide, and you'll be able to see the bacteria by magnifying your microscope to 40 times. Then you can count the bacteria in 10 to 15 of 1 over 400 squares. Then you can calculate the average size. The formula that we actually use to calculate this is total cell counted times 2 times 10 raised to the power 7 times dilution factor which you used. So the next one we'll be talking about is using the fluorescent dyes. Using dyes helps to magnify the cells that you have on a slide or probably on a petri dish. It's an important and unique method of calculating bacteria on a plate or on a slide. But this type of method does not count the bacteria that are viable or not. It's just like the former method we talked about, using direct cell counts. Okay, these are the advantages of this method. This method is actually fast and rapid. You can observe the morphology of the bacteria while counting the numbers. If you actually what do we call it, place your microscope well and you project it well, you can actually view well. The most of the limitations we have is that they discriminate between living and dead cells. Yes, this is a very important factor. You can't use this to count viable cells because most times in food we're trying to count number of viable cells that we're finding in this sample that can be able to cause disease. If you're trying to do that, you will not be able to use this method. So now we are moving to the viable plate count method. This method is called viable because it reveals the information of living bacteria. It's a very important method that is used, and in this method a very small volume 0.1 or 1 ml of your liquid containing the unknown bacteria is used. And importantly, we use the colony forming unit to calculate this in this method. A colony is a cluster of bacteria growing together, which you can obviously see on a petri dish. And this unit, to calculate this unit, bacterial cultures are added to the agar plate, often by serial diluting the original sample, and which might be too concentrated to count. I want to show a pictorial view of what I mean. Now, this is the first sample that you just diluted by 10 raised to the power 1, that's taking 1 ml and putting in 9 ml of water, 1 ml and putting 9 ml of diluent, taking 1 ml of your suspension containing your bacterial culture and putting in 9 ml of diluent, you would have this. Then as you continue to dilute, you can see from the plates that the concentration of microorganisms are beginning to reduce. So this is what we actually try to achieve using this method. And okay, let me do another explanation. As you can see now, this is from your original inoculum. You take 1 ml from your suspension plate containing your organism, and then you have to dilute it by adding this 1 ml to this plate, to this sample. Then from that you also add another 1 ml. This is what we call serial dilution, and you continue that way, continue